Hello everyone, welcome to a new session on Introduction to Bioinformatics. Here we discuss about the Human Genome Project. The genetic factors makes every person unique in every aspect, from the color of an individual's eyes to the functions of white blood cells. The most important thing that makes an individual's unique characteristics the hardest to understand is that it is all composed of 4 bases: adenine, thymine, guanine and cytosine in different patterns, the only thing that differ one human from other. This genetic code is found in every cell of the human body. In the 19th century, Gregor Mendel first discovered the foundations of inheritance, and it was the basis that has now made it possible for humans to learn more about the genetic code. Mendel's discovery has then turned into a multi-billion-dollar project known as the Human Genome Project (HGP). Now let us look back at a brief history about the Human Genome Project. The Human Genome Project began as a joint effort between the US Department of Energy (DOE) and the United States National Institute of Health (NIH). The Department of Energy's initial reason for entering the project was to gain a better understanding of the potential health risks that were involved in energy use and the production of energy, especially the risks involved with radiation. After 2 years of proposing the idea of sequencing the whole human genome, the NIH joined the effort. The foundations of the project were laid, and 2 years later, in 1990, the project had begun. The project was originally laid out to be a 15-year program that would have a budget estimated at $3 billion. Firstly, the Department of Energy and the National Institute of Health established 5 major goals for the Human Genome Project. The first goal is to identify all of the genes in human DNA. This goal is phenomenal when it is considered that there are an estimated 80,000 to 100,000 genes present in the human genome. The second goal of Human Genome Project is to obtain the sequences of 3 billion DNA base pairs that create the human DNA. The next 2 goals of Human Genome Project were to store all of the data on databases and create tools to analyze this information. The final goal of the project is to tackle the ethical, legal and social issues that may arise during the time it takes to complete the project. The completion time of Human Genome Project has been accelerated due to new advances in the technology. The new goals include having a working 90% draft sequence by the summer of 2000 and finishing the project by the year 2003. The finished project in 2003 would be a 100% high quality sequence of all of the base pair sequences of the human genome. The Department of Energy and the National Institute of Health have also stated that one of the highest priorities of the Human Genome Project is to not only complete the project, but to make all of the information available to the public. The early completion of the Human Genome Project does not come at a bargain price. The estimated budgets for 1999 alone are $89.8 million for the Department of Energy and $225.7 million for the NIH, bringing the grand total to $315.5 million for 1 year. The Human Genome Project has progressed faster than expected, but a race to be the first to complete the human genetic sequence has emerged and become very aggressive. For example, in 1998, the Perkin-Elmer Corporation and Dr. J. Craig Venter, head of the Institute for Genome Research, announced that they intended to start a new genomics company. The company's intent was to complete the sequencing of the genome within 3 years, that is 2001, making their completion date the earliest yet to be announced. They believe that their new company will be able to provide this information faster and more accurately than the current possible methods. The main aim of the Human Genome Project is the identification of genes. Our genetic makeup has involved with a large number of genes. The project aimed at the identification of 25,000 genes and creating a database for them. Other significant aim of the Human Genome Project involved determining the sequence of chemical base pairs. As a matter of fact, it is proved after repeated and trusted experiments that the DNA molecule consists of specified and well-defined sequence of 2 pairs of bases. Therefore, the Human Genome Project aimed at determining these particular sequences of nearly 3 billion base pairs that are responsible for the structure of DNA molecule. The other important goals aimed and achieved through this project involved the formation of proper and usable database for all these obtained information about the genes and the base pairs forming the DNA molecule. The scientists were of the view that if they succeed in getting all this information there by creating a database, it would mean that they will be able to solve the thousands of genetic issues associated with our life and in general in all sort of life. Now, let us study about the technical aspects involved in the Human Genome Project. Samples from many people were incorporated into a genomic library to use for Human Genome Project. The samples were taken from the sperm of males and the blood of females. Sperm taken from males for the Y chromosome that female cells lack. The donations of sperm and blood were strictly anonymous. The person donated their blood or sperm never knew that it was used in the Human Genome Project. The scientist who sequenced the genome also had no knowledge of the identity of the person. In addition to sequence the human genome, scientists have also worked on sequencing other species genomes to aid in the process of Human Genome Project. The genomes of Escherichia coli and Saccharomyces cerevisiae or baker's yeast have recently been completed. Researchers also studied the genomes of Drosophila and laboratory mice to aid in the project. Determining the 3 billion bases and approximately 100,000 genes of the human genome was accomplished by sequencing. The cutting DNA with restriction enzymes were used for sequencing. These enzymes cut the DNA at specific sites on the strand. The next step was to use gel electrophoresis to separate different sized fragments that might have been produced by different individuals when treated with the restriction enzyme. The gel allows different sized particles to move at different rates when an electric field is produced. The smaller fragments move more quickly down to the gel than the larger particles. The DNA can be amplified by either using a cloning vector such as a yeast artificial chromosome or a bacterial artificial chromosome, or by polymerase chain reaction or PCR to create a clonal library. The sequencing can begin after a clone library is achieved. Sanger method is one of the most popular methods being used for the sequencing of human genome. The first step of this method is to produce fragments that each end in a different base. These fragments are then combined with replication reagents and replicated. One batch of fragments producing only fragments that end with adenine, one batch only ending in guanine, one in cytosine and one in thymine. The fragments are then sorted by gel electrophoresis to allow determination of the sequence of DNA. The development of new technologies were constantly being funded for the overall completion of the project, not only faster but also cheaper. Systems have now been developed that contain greater number of lanes and lower run times, for example, capillary array electrophoresis or CAE. This system involves the use of a laser to read 96 capillaries at one time. The time is measured that it takes for each of the fragments to reach the laser detector region. Other new technologies have also been developed such as shotgun sequencing, directed approach sequencing and sample sequencing. Now, let us look at the concerns of the Human Genome Project. There were a variety of legal and ethical concerns behind the completion of the Human Genome Project that varies in subject from clinical applications to commercialization to privacy. The Department of Energy and the National Institute of Health estimated that these concerns would play a big role in the Human Genome Project. They have developed a committee to deal with these issues. This group considered the access to genetic tests, fairness of use of information, employment and insurance situations, confidentiality and storage of information, and public education. One of the most controversial topics surrounding the Human Genome Project was the issue of commercialization of the products resulting from the completion of the project. Clinical applications of the project were also another major concern of the public. These applications may be able to aid the human population greatly in terms of health care. For example, after a mutation has been identified, doctors can then begin to work on genetic screening processes and allow them to predict the possible onset of a genetic disease. However, the main goal of clinical applications is gene therapy. Gene therapy would provide a treatment to those who are suffering from a disease. Eventually, gene therapy may even help doctors find a cure for many genetic diseases. Gene therapy is already being tested for genes that have been identified and been tested for diseases such as cystic fibrosis and Duchenne muscular dystrophy. One of the downsides to these clinical applications is the impact of this information might have on human lives. Another ethical and social related concern of the Human Genome Project is the issue that deals with fairness and confidentiality. The completion of the Human Genome Project makes the genetic screening may become a lot more common than previous. A majority of the public is concerned with the impact these tests could have on their employment opportunities and insurance premiums. In a survey conducted by the Department of Energy and NIH Working Group on Ethical, Legal and Social Implications of the Human Genome Project, nearly 7 out of 10 Americans are concerned that genetic information may be used against them. In 1995, a set of guidelines was written to advise lawmakers to try and prevent insurance companies from raising rates or deciding eligibility based on genetic information. Unfortunately, these were merely guidelines and not a law. Luckily, in 1996, an act was passed to prevent the increase of rates for group health care because of genetic status. This act does not ensure privacy. However, many other acts have been proposed to Congress but are still pending. Genome sequencing in Human Genome Project. The sequencing of the human genome involves figuring out the order of all 3 billion bases that make up the DNA. This was one of the major challenges of the Human Genome Project. The process, which was automated for the project, includes several steps which is given in the following figure and explained as follows. 1. The chromosomes are broken into shorter pieces of DNA. 2. Each single stranded short piece was used as a template for the enzyme DNA polymerase to make a new complementary piece of DNA. The polymerase can add one of 2 types of bases to make the new piece of DNA. It can add a normal base such as A, adenine, G, guanine, C, cytosine or T, thymine, or it can add a version of one of these bases that stops replication because the modified base cannot have another base added after it. The right concentration of these bases along with normal bases will result in many different strands of DNA being made, each a different length. 3. The next step is to separate the newly made strands by gel electrophoresis based on their size. Shorter strands travel faster through the gel than longer strands. If the second step was successful, an original strand that was 10 bases long will result in 10 different strands separated out by size on the gel, each one base longer than the next. Fourth, the base that is at the end of each strand has a fluorescent tag attached, a different color for each of the bases. These colors can be seen with a fluorescent microscope, and the sequence of the DNA is determined by the sequence of the colors that come up on the gel. For example, a sequence of bands that are red, green, green and blue might mean that the 4-base strand was made up of adenine, guanine, guanine and cytosine, or AGGC. 5. A computer reads the colors and determines the sequence, which it then combines with all of the other sequences to eventually make up the whole genome. This sequence information is then submitted to larger public databases so that other researchers can use it and add new information to it. These human genome sequences do not represent any one person's genome. They are intended as a starting point for broad comparisons across humanity. The knowledge can be applied to everyone because all humans share the same basic set of genes as well as the development and maintenance of their biological structure and processes. Based on Human Genome Project, human genome sequence can be classified into different groups depending on the overall sequence composition and structure as shown in the following figure. There is an estimated 20,000 to 25,000 human protein coding genes. The protein coding sequences account for only a very small fraction, approximately 1.5% of the human genome, and the rest is associated with non-coding RNA molecules, regulatory DNA sequences, lines, signs, introns and sequences for which as yet no function has been determined. Based on Human Genome Project, the content of human genome is commonly divided into coding and non-coding DNA sequences. Coding sequences, exons, are transcribed into mRNA and in which the corresponding mRNA molecules are translated into a polypeptide chain. Every 3 nucleotides termed a codon in a protein coding sequence encodes one amino acid in the polypeptide chain. In some cases, it may either map a given codon to a different sequence or may use different codons more or less frequently. Therefore, some protein coding sequences may be optimized for use in a particular chassis. The protein coding sequences begins with a start codon, usually ATG, and end with a stop codon, usually with a double stop codon TAA, TAA. Although protein coding sequences are often considered to be basic parts, in fact, proteins coding sequences can themselves be composed of one or more regions called protein domains. Thus, a protein coding sequence could be entered as a basic part or a composite part of 2 or more protein domains. The N-terminal domain of a protein coding sequence is special in a number of ways. First, it always contains a start codon spaced at an appropriate distance from a ribosomal binding site. Second, many coding regions have special features at the N-terminus such as protein export tags and lipoprotein cleavage and attachment tags. These occur at the beginning of a coding region and therefore are termed head domains. A protein domain is a sequence of amino acids which fold relatively independently and which are evolutionarily shuffled as a unit among different protein coding regions. The DNA sequence of such domains must maintain in frame translation and thus is a multiple of 3 bases. Since these protein domains are within a protein coding sequence and are called internal domains. Certain internal domains have particular functions in protein cleavage or splicing and are termed special internal domains. Similarly, the C-terminal domain of a protein is special, containing at least a stop codon. Other special features such as degradation tags are also required to be at the extreme C terminus. Again, these domains cannot function when internal to a coding region and are termed tail domains. Non-coding sequences are the sequences that are not used to encode a protein. Even though some non-coding DNA sequences are transcribed into functional non-coding RNA molecules like transfer RNA, ribosomal RNA and regulatory RNAs. In addition to the non-coding RNA molecules that are encoded by discrete genes, the initial transcripts of protein coding genes usually contain extensive non-coding sequences in the form of introns, 5' untranslated regions or 5' UTR and 3' untranslated regions or 3' UTR. Within most protein coding genes of the human genome, the length of the intron sequence is 10 to 100 times the length of exon sequences. The non-coding region is of different types: repetitive introns, regulatory, etc. Repetitive DNA sequences. Repetitive DNA sequences comprise approximately 50% of the human genome and are of two types, tandem and interspersed. About 8% of the human genome consists of tandem DNA arrays or tandem repeats, low complexity repeat sequences that have multiple adjacent copies. The tandem sequences may be of variable lengths from 2 nucleotides to tens of nucleotides. These sequences are highly variable even among closely related individuals and so are used for genealogical DNA testing and forensic DNA analysis. Repeated sequences of fewer than 10 nucleotides are termed microsatellite sequences. Among the microsatellite sequences, trinucleotide repeats are of particular importance as sometimes occur within coding regions of genes for proteins and may lead to genetic disorders. Telomeres or the ends of linear chromosomes end with a microsatellite hexanucleotide repeat of the sequence. Tandem repeats of longer sequences or arrays of repeated sequences 10 to 60 nucleotides long are termed as mini satellites. Interspersed DNA sequences. The interspersed DNA sequences are of different types. Transposons are segments of DNA that can move around to different positions in the genome of a single cell. In the process, they may cause mutations and increase or decrease the amount of DNA in the genome of the cell, and if the cell is the precursor of a gamete, in the genomes of any descendants. These mobile segments of DNA are sometimes called jumping genes. There are two distinct types: class 1 transposons, class 2 transposons. Class 1 transposons are also called retrotransposons that first transcribe the DNA into RNA and then use reverse transcriptase to make a DNA copy of the RNA to insert in a new location. Class 2 transposons consists of DNA that moves directly from place to place. 50% of the human genome consists of retrotransposons and are of two types, signs and lines. Lines are also known as long interspersed elements and are 19% of the human genome. Signs are short DNA sequences around 100 to 400 base pairs that represent reverse transcribed RNA molecules originally transcribed by RNA polymerase III, that is molecules of tRNA, 5S rRNA and some other small nuclear RNAs. Introns are the non-coding sections of a gene that can transcribe into the precursor mRNA sequence, but removed by RNA splicing during the processing to mature messenger RNA. Some introns appear to have significant biological function, possibly through ribozyme functionality that may regulate tRNA and rRNA activity as well as protein coding gene expression, evident in hosts that have become dependent on such introns over long periods of time. Also, there are genes that are similar to normal genes but non-functional called pseudogenes. In any study of molecular evolution, it is necessary to compare and contrast genes from a variety of organisms to gauge how the organisms have adapted to ensure their survival. Pseudogenes are vitally important since they provide a record of how the genomic DNA has been changed without such evolutionary pressure and can be used as a model for determining the underlying rates of nucleotide substitution, insertion and deletion in the greater genome. Applications of the Human Genome Project. The current and potential applications of the Human Genome Project are numerous. With the sequence of the majority of human genes now established, recent work has been focused on the function of genes and how changes in the sequence relate to health and disease. The description of the genome has led to interest in other omes such as the proteome and the metabolome. As we find out more about human genes and how they work, we will be able to develop new ways to diagnose and treat more diseases. The Human Genome Project has already fueled with the discovery of more than 2000 disease genes. As a result of the Human Genome Project, today's researchers can find a gene suspected of causing an inherent disease in a matter of days rather than the years it took before and the genome sequence was in hand. There are now more than 2000 genetic tests for human conditions. These tests enable patients to learn their genetic risks for disease and also help healthcare professionals to diagnose disease. At least 350 biotechnology based products resulting from the Human Genome Project are currently in clinical trials. Having the complete sequence of the human genome is similar to having all the pages of a manual needed to make the human body. The challenge now is to determine how to read the contents of these pages and understand how all of these many complex parts work together in human health and disease. One major step towards such comprehensive understanding was the development in 2005 of the HapMap, which is a catalogue of common genetic variation or haplotypes in the human genome. In 2010, the third phase of the HapMap project was published with data from 11 global populations, the largest survey of human genetic variation performed to date. HapMap data have accelerated the search for genes involved in common human diseases and have already yielded impressive results in finding genetic factors involved in conditions ranging from age-related blindness to obesity. The tools created through the Human Genome Project continue to underlie efforts to characterize the genomes of important organisms used extensively in biomedical research, including fruit flies, roundworms and mice. National Institute of Health's Ethical, Legal and Social Implications program has become a model for other research efforts seeking to address ethical issues in a proactive manner. With a drastic decline in the cost of sequencing whole exomes or genomes, groundbreaking comparative genomic studies are now identifying the causes of rare diseases such as Kabuki and Miller syndromes. Much work still remains to be performed. Despite many important genetic discoveries, the genetics of complex diseases such as heart disease are still far from clear. Pharmacogenomics is a field that looks at how genetic variation affects an individual's response to a drug. Pharmacogenomic tests can already identify whether or not a breast cancer patient will respond to the drug Herceptin, whether an AIDS patient should take the drug Abacavir, or what the correct dose of the blood thinner warfarin should be. The Cancer Genome Atlas. The Cancer Genome Atlas is an ambitious new initiative which aims to identify all the genetic abnormalities seen in 50 major types of cancer. Based on a deeper understanding of the disease at the genomic level, we will see a whole new generation of targeted interventions, many of which will be drugs that are much more effective and cause fewer side effects than those available today. National Institute of Health supported access to high throughput screening of small molecule libraries, which will provide academic researchers with powerful new research probes to explore the hundreds of thousands of proteins believed to be encoded by the approximately 25,000 genes in the human genome and will provide innovative techniques to spur the development of more new effective types of drugs. NIH is striving to cut the cost of sequencing an individual's genome to $1,000 or less. Having one's complete genome sequence will make it easier to diagnose, manage and treat many diseases. Individualized analysis based on each person's genome will lead to a powerful form of preventive, personalized and preemptive medicine. By tailoring recommendations to each person's DNA, healthcare professionals will be able to work with individuals to focus effort on the specific strategies from diet to high-tech medical surveillance that are most likely to maintain health for that particular individual. The increasing ability to connect DNA variation with non-medical conditions such as intelligence and personality traits will challenge society, making the role of ethical, legal and social implications research more important than ever. Let us now summarize what we have learned in this session. So far, we have had amazing success with the Human Genome Project. The only problem is that the technology does not exist that we need. Perhaps the ultimate problem is that we keep on reaching new conclusions which require so much thought and new technologies. There are, however, considerable bonuses to the Human Genome Project. Although we do not know if we need to map out the entire human genome, we do know that it is a wealth of knowledge. For example, the roundworm that we decoded had a huge amount of brand new genetic information that we had never seen before. Because we can now start to compare the genes of various species, we get a better and bigger organization and thus how we all linked together, which should give us a very strong creation theory. Because of this, the Human Genome Project must continue in order to expand our knowledge. The Genome Project has already had a profound impact on biomedical research as evidenced by the isolation of a number of genes associated with important diseases such as Huntington's disease, amyotrophic lateral sclerosis, neurofibromatosis type 1 and 2, myotonic dystrophy and Fragile X syndrome. Genes that confer a predisposition to common diseases such as breast cancer, colon cancer, hypertension, diabetes and Alzheimer's disease have also been localized to specific chromosomal regions. Here are a few questions for you to work out. 1. What is the role of Department of Energy in Human Genome Project? 2. What are the applications of Human Genome Project in medical science? 3. Find the databases that provide the Human Genome Project data. 4. What is the ethical, legal and social implications or ELSI research program? What is HapMap? Here I can suggest a few books and websites for your reference. 1. The Human Genome Project and Future of Healthcare, edited by Thomas H. Murray, Mark A. Rothstein, Robert F. Murray Jr., printed by Indiana University Press, 1996. Second one, Understanding the Human Genome Project, written by Michael Angelo Palladino, published by Pearson, Benjamin Cummings in the year 2006. The Human Genome Project by James Torriello, published by Rosen Publishing Group, 2003. Fourth one, The Genome Project, Why the DOE, obtained from www.ornl.gov/HGMIS. Fifth one, Human Genome Project information about the Human Genome Project, obtained from www.ornl.gov/HGMIS. Human Genome Project information about the Human Genome Project can be obtained from the website www.ornl.gov/HGMIS. hgmis/about.html. Thank you for watching this program. See you next time. Until then, bye.